Welcome back. So this video is going to go through the 3 types of mutations that are specifically mentioned in the study design. So this is a huge dot point. We break it into 4 lessons, just so you have an idea that you're not expected to just be able to recall all of this the same way that other dot points are. So there are 3 different types of mutations we're going to go through. We're going to go through point mutations, frame shift mutations, and block mutations. So you'll see on page 3 of your booklet is there's a table there. So the first thing you got to do is put a diagram in, then we've got to put a description, and then we're going to talk about how severe the implications of that mutation might be. So my suggestion is this video is probably going to take a little bit longer than normal because you'll need to pause, draw something in your book, and then come back and watch a little bit more. You might need to watch it a couple of times just to get your head around the 3 different types of mutations. But before we waste any more of your time, let's get going. So there are 2 parts of this course. The second part here, this chromosomal abnormalities, is not in 2020. Yes, so there will be a video added later on for next year's cohort. However, at this stage, you don't need to understand chromosomal abnormalities. So we're just going to look at gene mutations. All right, so sometimes there's a problem with the structure of the original gene and the protein will be different. Most of the times it's bad, but sometimes it's good. And what we're hoping for is to drive evolution and to drive this change over time is we need some of these good mutations to occur. So there are 3 main types that we can talk about: a point mutation, a frame shift mutation, and a block mutation. So let's get started on what a point mutation is. So there are 3 different types of point mutations that I want us to go through. Sorry, girls, the 3 different types of point mutations. But before we do that, let's just think about what a point mutation might be, having a look at this diagram. So having a look at this diagram, the red is showing me the changes. How many bases are changing each time? Hoping you're looking at it and you're going, well, in a silent mutation, in this first one, only the T has changed at the end here. So here is the original. We can see that in a silent mutation, we've got the T changed. In a nonsense mutation, for this example, we've got the A change, and the missense, we've got 2 different types of missense mutations here. And they're that. So a point mutation is a change in a single DNA base pair. And what's important to acknowledge is that where that change happens depends on, will implicate what the effect of that mutation is. So I'm going to start off with our least severe mutation, and that's a silent mutation. So give me 1 second. I'm just going to pull up the codon table to help us with this. Good, I'm all ready now. So I didn't put this one in your book, but if you have access to an old copy of your codon table, you might need to pause, go and find it, and bring it back. So we talked about in area of study 2, the fact that the genetic code was universal. In this area of study, we actually need to focus on the fact that the genetic code is, if I can spell, that is. The genetic code is, and I'm going to put it down here so it's a bit easier to see, genetic code is redundant. Now what that means is that you can have a change in that genetic code, like this one here has changed, and it did not change the amino acid. So here, remember, here is the original, and if you don't change the amino acid, we call that a silent point mutation. So that's our first time. Do you think it's going to have any kind of impact on the protein? If we haven't changed our amino acid, this has no impact on the protein. So that's why it's called silent. Yeah, so this is one of the types of mutation. So a point mutation, remember, is a change in one DNA base pair. Good. All right, so that might be any of these ones here where the amino acid has lots and lots of different codons. If you had a mutation in that third base there, then that means that it would be a silent mutation. So if it changed from CCU to CCC, then it wouldn't change the amino acid that it coded for. It would still code for proline, which is this one here. If I changed from, let's have a look. If I change from UCU to UCA, both of them, if I just change that last base there, they would all code for serine. So that mutation would be an example of a silent point mutation. So it has no effect on the individual, no impact on the protein. The next type of mutation, I'm actually going to jump to the far right. I'm going to talk about these guys. So that's 1 is silent, 2 is missense. So missense is when you change a base and it changes the amino acid that it codes for. So you can see here that when this middle base changed to a C in the DNA, it changed the mRNA, and that there changed the amino acid. Now this could, as a result, cause changes in the primary structure and therefore the protein. This could affect the functioning of that protein. It depends. So some of them, you can see here that arginine is actually very similar in structure to lysine. So it's perhaps if it changed like that, it wouldn't change your protein. However, if it changed to this case here, this threonine example, so it changed from lysine to threonine, then what's going to happen is you're going to get a big change in shape. It's not going to bond the way that it used to, and so therefore you're likely to end up with changes in primary structure, secondary structure, and tertiary structure. So if you're looking at me and going, oh God, what does she mean by primary, tertiary, and secondary and tertiary structure? It's time to go back, have a look at your protein notes from Unit 3, because this is directly linked in to what we did in Unit 3. The final type of point mutation I'm going to go through here is a nonsense mutation. So this one here is where you change one base. Again, we're just changing one base, but instead of it coding for a different amino acid, it now codes for a stop codon. Now what that means is that your primary structure is going to end earlier. So this could have a very large effect if at the beginning of the chain, so the beginning of your polypeptide chain. If it happens at the end of your polypeptide chain, then it might have a smaller effect. So overall, your severity for your point mutations can vary from 0 impact with a silent mutation, some impact with a missense mutation, depending on how bad it is. We could have a large impact with a missense mutation where you change a whole amino acid, or it could even have a larger effect if you get a nonsense mutation, which is where you stop your protein early or stop your polypeptide chain early. So there's that does kind of depend there. So there's one more little picture just to show you some different types of point mutations. You can get what we call a substitution mutation. This is where you're not deleting something, but we're just changing the B to an F in this case. Now you can see it does change the meaning. You've also got an insertion mutation where if you put in another letter, it becomes something different as well. So you can see in this example that by changing little parts of it, then you end up with different effects. So the original word was beast. By substituting the B for an F, it becomes feast. You could insert something, you could make it breast. You could delete an A. You could have a deletion mutation and it would be best. Or you could even have this thing called an inversion where you switch around the last two parts of that and it becomes beats. Pretty cool. All right. So the next type of mutation we're going to talk about is a frame shift mutation. So a frame shift mutation is essentially, you've got to think about reading the DNA in threes in those codons, yes? And each codon has a particular order. If you do something like an insertion or a deletion, then what you can end up with is actually a shift in the way the whole protein chain is red. So the whole mRNA or DNA sequence is red. So you can see here is the original DNA code. It starts to read CAT, TCA, CAC, ETA, so on and so forth. So if it reads in that order, it codes for these amino acids. However, if you frame shift and you either say you added in a base at the beginning, then all of a sudden we start reading different sets of 3. Now, if you start reading different sets of 3, can you see how every single amino acid after there has changed? So this one starts off with his, this one starts off with ill. This one then goes to serine, then this one's changing to his. Can you see how the entire sequence is different? Yeah, and if it's different as we move down there, that's going to have a large impact on my protein. So the 2 different types of frame shift mutations that we're going to talk about are things called an insertion or a deletion. So a frame shift could be of a single base or a kind of chunk or section of base pairs. So this is why I talk about frame shift as a separate type of mutation. So it could be a subset of a point mutation, or it could actually be a subset of our next section, which are called block mutations. So an insertion is where you insert, funnily enough, 1 or more DNA base pairs that cause the codons to be read differently. Okay, the next one is a deletion. This one is where you remove 1 or more DNA base pairs that cause the codons to be read differently. So you can see here that so long as you either insert or delete, you're going to change all the amino acids. Hang on. So a frame shift mutation changes all amino acids after the mutation. So this one's more severe usually than what we were talking about with the point mutations, with the fact that you could have a silent point mutation, you could have a missense point mutation. The nonsense point mutation and a frame shift mutation tend to sit on the same kind of level because they're affecting everything after the change. Now, the final type of mutation is virtually, it can incorporate all of that stuff that we've learned. So you can have deletions, you can have duplications, or you can have insertions. So duplications or insertion. But instead of it being just 1 base pair, this is more than 1 base pair. So usually it's like a whole chunk of DNA that will either get deleted from a chromosome, it could get a whole chunk of DNA that gets inserted into a chromosome, and then there are these 2 last ones down here that we haven't really talked about. You could take this section, this green section of DNA, and flip it around. Yeah, if you flipped it around, then you get what we call an inversion. So say the DNA got cut or it got twisted, because remember DNA are these long strands, so maybe it folded back on itself and twisted around, and you end up with a part of a chromosome that's in the wrong direction. That means that whole section of chromosome, when your RNA polymerase comes along to read it, is going to be read incorrectly. The final one is a translocation, where this part of the chromosome, sorry, that's the wrong color, let's use this color. This last little blue part of the chromosome probably came from another nearby chromosome. So it's where you get part of another chromosome coming over and attaching itself to this. Now, this is a really big video. There's a lot of concepts here. I'd like to unpack them further with you, but this is a nice little summary for you to come back and go, what is the difference between these different types of mutations?